The background of this study is the global market of probiotic products is growing rapidly, and as we all know that this product must go through several assessments to make sure their safety and health benefits of probiotic strengths. However, safety issues of probiotic products have been reported frequently in recent years. So, in this study, they came out with the idea to do a whole genome analysis as an expected method that could resolve this problem. In this study, 10 strains were isolated from 7 commercial probiotic products, and several tests were performed, such as strain-level identification, evaluate the presence of antimicrobial resistance and virulence factors genes, and also evaluate the genomic instability of the isolates. Comparative genome analysis also have been done for the Enterococcus strains.
So I will explain the method step by step. Firstly, 7 probiotic products were dissolved in 20 ml PBS for mixing and culture on different mediums such as TSA agar, MRS agar, and MC agar under anaerobic condition. Then the total DNA was extracted using purigine yeast vacuum, and quality and quantity were evaluated by agarose gel. After the whole genome sequencing was performed on both Illumina High Sequence Extend platform and Oxford Nanospore Technology (ONT), for Illumina sequencing, about 350 base pair DNA library were prepared, while Smart sequencing, about 20 to 30 kb only were prepared using Oxford Nanospore Technology. Then, ANI was used for genome-wide comparison for bacteria identification, and it was calculated by FastANI. The web-based platform that was used for strain typing is pubmlst.org. For identification, the clustered regulated interspace short palindromic region were identified using CRISPR-Cas finder tools. Putative prophage sequence were identified using Prophage Hunter, and for bacteria insertion, sequence using IS-Finder. Next, AMR gene were identified by BLAST-P with comprehensive antibiotic resistant database card, while putative virulence were identified by BLAST against virulent factor database VFD, and gene encoding toxic metabolite sequence using UniProt. The core gene of both strains, which are E. faecium and E. faecalis strain, were obtained using BLAST 2.2 and OrthoMCL version 1.14, and the phylogenetic analysis were conducted using IQ3. Lastly, the correlation between IS element and toxic metabolite was calculated by R statistical platform, and corresponding figure generated by using FitMap package.
Firstly, we talk about isolation and identification. Usually, this mislabeling of probiotic isolates can caused by inaccurate taxonomic identification. So, this identification of bacterial isolates and mislabeling is very important in safety of probiotic products. Using various tools, we 10 bacterial isolates were identified, and among the 10 bacterial isolates, 3 were inconsistent with their corresponding product labels based on whole genome identification. Next is identification at strain level. 2 methods were used. One is strain typing by single nucleotide polymorphism distance. Results showed that only TKJ6A isolate has minimum SNP distances with the probiotic strain Bifidobacterium animalis subspecies lactis B420, which are similar and can meet the threshold. For other probiotic product isolates, the minimum SNP distances are all more than or equal to 40 base pair and not enough for strain typing. The second method is strain typing by MLST based on whole genome. Only 3 probiotic product isolates can be assigned to strain type. The first safety assessment is antimicrobial resistance. From the result, the antimicrobial resistance gene were identified in the genome of 3 isolates. Next safety assessment is for virulence factor. Based on the result, virulence factor were detected in 3 isolates and no genes encoding recognized virulence factor were found in Bifidobacterium and Lactobacillus isolates. Followed by the safety assessment for toxic metabolites. From the result, genes associated with biogenic amine synthesis found in both Enterococcus isolates. Key genes for D-lactate synthesis were identified in 4 lactobacillus isolates and 1 lactobacillus isolates contain key genes associated with nitro compound production. These IS elements from 14 families found in genome of 9 isolates, and the result indicates specific IS elements at strong correlation with the virulence factor. Moreover, in the genome of Lactobacillus helveticus, a hypothetical gene was found between 2 IS elements and a glycosyl transferase gene was located between another 2 IS elements, indicating potential genetic instability. The next mobile genetic element is multidrug resistant transposon was identified in the genome of Enterococcus faecalis. The next mobile genetic elements are prophage and plasmids. Active prophage genes were identified in the genome of 4 isolates, and plasmid characterization of 10 probiotic product isolates shows that 7 isolates contain epigenetic elements. Next is CRISPR-Cas system and genomic islands. This CRISPR-Cas system is an adaptive immune system of bacteria which protects the bacteria from invaders. If the bacteria contains no CRISPR-Cas system, the gene acquisition from bacteriophage will increase in the bacterial strain. So, the CRISPR-Cas system were identified in 6 isolates, and 3 isolates contain orphan CRISPR arrays without Cas genes. Moreover, genomic islands were not found in any isolates. Next is the analysis of correlation between antimicrobial resistance and virulence factor genes with the mobile elements. From the result, no antimicrobial genes found in the mobile genetic elements and no strong correlation was found between antimicrobial genes and insertion sequences. For virulence factor, results show that virulence factor has high correlation with the mobile elements. For this comparison, phylogenetic analysis was carried out with 2 isolates from the genus Enterococcus. First analysis was carried out for Enterococcus faecalis. From the phylogenetic tree, we found that the Enterococcus faecalis was clustered with pathogenic strain. From the phylogenetic analysis, we found that the Enterococcus faecium was clustered with non-pathogenic strains.
Whole genome SNP distance used to assess the genetic relatedness among bacteria isolates. The major restrictive factor for strain level identification of PPS employing SNP distance is the limited number of published genomic sequences as comprehensive genome database. It is constructed for safety evaluation, strain typing, and property right protection, especially in probiotic area. In QPS criteria, probiotic assessment includes identity, body of assessment, safety concern, and antimicrobial resistance. Ideally, probiotic should not carry intrinsic antimicrobial resistance. PPS contain AMR genes that can potentially render the effectiveness of antibiotics. Although AMR seems to be intrinsic, but with multi-drug resistant genes may potentially induce safety issues when they obtain external virulence genes with very small probability. VFs related to capsule or biofilm formation and bacterial adherence facilitate probiotic survival in gastrointestinal tract by strengthening the acid resistance and intestinal colonization. lactobacillus isolates can produce D-lactic acid, while 2 Enterococcus isolates can produce natural compound. Maybe these toxic metabolites produced under certain condition in specific host. The absence of plasmid active prophage genes or transposon indicates its genome is stable. The absence of Cas system may increase the likelihood of AMR acquisition from bacteriophages or plasmids. That's the reason for the presence of embedded active prophage genes sequences, multi-drug resistant associated transposon, and virulence associated plasmid in PPS. 2 Enterococcus isolates potentially display drug resistance, virulence factors, toxic metabolites, and genetic instability.
First, the government should stringent the rule and regulation for the probiotic drink or any type of drink that contain probiotic in their product. Because most of the probiotic drinks or product in the market right now have a misleading information with the incorrect content of the probiotic inside it. By using the whole genome sequencing method, we can make a better safety evaluation for the health purpose. From the result of the whole genome sequencing, we can recognize multiple types of harmful substance from the drinks. First, we can identify the misleading and the incorrect content by using strength typing. Then, from the whole genome sequencing, we can identify the genome instability involving transposon, which also lead to any antibiotic resistance. Thirdly, from the result also, we can identify the region of the antimicrobial resistant genes, virulent factor, and any toxic metabolite in the strain that extracted from the market probiotic drinks. Then the probiotic can be analyzed also to compare it, is it pathogenic or not pathogenic towards our body. This show how important the whole genome sequence method to become the best approach for the probiotic drink safety evaluation.